Hello guys, I'm gonna do a series of videos on protein synthesis. In this part, I'm gonna talk about the translation machinery, the transfer RNAs and ribosomes, and I'm gonna talk about the Wobble hypothesis. But let's start by a general overview on protein synthesis. Protein synthesis is the process in which cells make proteins. It occurs in two steps: transcription and translation. Transcription is the process in which the DNA sequence of a gene is copied or rewritten in the form of mRNA. Translation is the process in which the sequence of nucleotides in the mRNA is read and decoded to a sequence of amino acids building a protein. Now let's review the difference between DNA and RNA. First, DNA is a double-stranded molecule, while RNA is a single-stranded molecule. Second, the sugar in DNA is the deoxyribose, while in RNA it's the ribose. Third, the nitrogenous bases in DNA are ACTG, while in RNA they are ACUG, which means there is no thymine in RNA but uracil. Now for the difference between mRNA and proteins. Protein is made of a sequence of amino acids, while mRNA is the sequence of nucleotides, and each three nucleotides form a codon. During translation, codons of mRNAs are read in the 5' to 3' direction, and they are translated into amino acids, forming a polypeptide chain synthesized from NH2 to COH terminus. To start with mRNA, each three bases form a codon that specify an amino acid according to the genetic code shown on the right. The genetic code is composed of 64 codons. The codons are read from 5' to 3'. Let me show you how. Let's take for example the start codon AUG. The first letter is A, the second letter is U, the third letter is G. This codon codes for the amino acid methionine, abbreviated as MET. Out of these 64 codons, only 61 codons represent 20 amino acids, and the remaining 3 represent the stop codons UAA, UAG, and UGA. So like I said previously, when a protein is translated from mRNA, it is done from N-terminus to C-terminus, where the new amino acid is added to the carboxyl end of the growing peptide chain. Or in other terms, the N-terminus of the new amino acid is linked to the C-terminus of the protein chain, and so on. Now, before I start detailing the protein synthesis, you should know that this process should be fast enough to meet a cell's needs and should also be accurate. It has been estimated that during translation, one amino acid could be misincorporated every 10,000 amino acids. Now let's get into details of protein synthesis, starting by the requirements of this process. First, we need an mRNA that contains the codons. Second, we need transfer RNAs that recognize the codons with their anticodons and carry the corresponding amino acid. Third, we need ribosomes that are the translation site. And finally, amino acids are needed to make proteins. In this part, we will see how the cell uses these ingredients to synthesize new proteins inside ribosomes. So let's talk more about these ribosomes. They are made of 3 ribosomal RNA and 50 proteins that form the small and the large subunit. There is 4 binding sites located on the ribosome, 1 for the messenger RNA and 3 for the transfer RNA sites labeled A, P, and E. The A site, called the acceptor site, binds to the tRNA holding the new amino acid. P site, called the peptidyl site, binds to the tRNA holding the growing peptide chain. And E site, the exit site, holds the deacylated tRNA, the tRNA that doesn't hold an amino acid, before it leaves the ribosome. Ribosomes are often referred to as catalytic RNA or ribozymes. To remind you, what are ribosomes? They are RNA enzymes, which means that they are RNA molecules that have the ability to catalyze enzymatic reactions. These ribosomes exist in the large subunit of the ribosome, and they catalyze joining amino acids together to form protein chains. tRNA are adapter molecules that bind to a specific codon by Watson and Crick base pairing, which means A pairs with U, C pairs with G, and brings with it a specific amino acid. You all know that there are 61 codons that synthesize amino acids. Therefore, there must be 61 tRNAs, each having different anticodons. But experiments showed that the total number of tRNAs is less than 61. So, there is at least one tRNA for each of the 20 amino acids, and this is the Wobble hypothesis. Let's take phenylalanine as example. There is two codon, UUU and UUC, that code for phenylalanine. Don't forget that we read codon in the 5' to 3' direction. But what do these codons have in common? Yes, the first two letters. It appears that UU specifies phenylalanine and that the third letter is not important. Phenylalanine tRNA with the anticodon 3' AAG 5' recognizes 5' UUC, showing a normal base pairing. But not only this codon, it also recognizes UUU codon with an unconventional base pairing where G of the anticodon base pair with U. The violation of the usual rules of base pairing at the third nucleotide of a codon is called Wobble. The first two bases, UU of an mRNA codon, strongly pairs with the corresponding bases of the anticodon AA, according to Watson and Crick base pairing. The third position base in codon and the first in anticodon located at the Wobble position doesn't bind as tightly as the first two bases, but it's possible. G could pairs with C or U, according to Wobble rule. Because of this flexibility, a cell doesn't need 61 different tRNAs, but less, to code for 20 amino acids. Not only that, but also certain tRNAs have the unusual nucleotide inosine in the first anticodon position. Because inosine can base pair with A, U, or C, according to Wobble rule that you can find in the table below, a tRNA containing the unusual base I can recognize 3 different codons. This result was obtained in an experiment done on yeast alanine tRNA, which has the anticodon 3' CGI 5', and it was found to bind to 3 codons, GCA, GCU, and GCC, as you can see in the figure. So, if we have an A in the 5' base in anticodon, this A will base pair with U in anticodon. If we have a C in the 5' base in anticodon, it will base pair with G in codon. If we have G, it will base pair in U or C, as we saw in the phenylalanine example. U in anticodon will base pair with A or G, and I will base pair with A, U, C, like the experiment we just gave. So, the first base of an anticodon determines whether a particular tRNA molecule reads 1, 2, or 3 kinds of codon. C or A will read 1 codon. U or G will read 2 codons. I will read 3 codons, like the example of alanine tRNA. Let's take a look at the structure of the transfer RNA. It's a single chain tRNA with cloverleaf structure containing 73 to 93 ribonucleotides. They contain unusual bases such as dihydrouridine and pseudouridine derivatives of uracil, pseudouridine being the most abundant RNA modification. Inosine is a derivative of adenine, methylcytidine is a derivative of cytosine, and methylguanosine derivative of guanine. The importance of this unusual bases is that they prevent the base pairing. In this way, they become accessible for other interactions, allowing the folding of the secondary structure into the tertiary structure, and it adds a hydrophobic character to some regions of the tRNAs, stabilizing the tertiary structure, thus protecting the tRNA from degradation. The secondary structure of most tRNA is composed of the following: 5' terminal phosphate group, it's usually a phosphorylated guanine. The acceptor stem on the 3' end that contains cytosine adenine sequence, the CCA sequence, which is used to attach the amino acid on the 3' OH of adenine. The D arm, also known as the DHU arm, that contains dihydrouridine, the anticodon loop that contains the anticodon complementary to the codon of mRNA, a variable loop, and the T-psi-C loop that contains unusual bases such as T, the ribothymidine, and Psi, the pseudouridine. In the 3 dimensions, tRNA adopts an L shape with the acceptor end on one end and the anticodon on the other end. As you can see, not all the nucleotides are paired, especially those in the loops, but we can say that half of them are base paired to form double helices. So the tRNA is a single-stranded molecule that shows significant intracellular base pairing, giving it its characteristic 3-dimensional shape.